So today's lecture is going to cover 21.5, which is alveolates, and 21.6, which are the Rhizarians. These two groups are the A and the R as part of the SAR supergroup. So we're going to start by talking about the alveolates. As I told you the first day when we introduced this, that we're going to discuss some of the characteristics. We're going to focus on some of the more common or well-known protists in that group. And then later on, you're going to do a little research on your own on one of these that's been assigned to you. So alveolates, when I see that word, I think of alveoli that are in the lungs. And if you look at the word alveolate, when you take alveolus from it, it means sac. And so basically these are little sacs that are under the plasma membrane. And in the lungs, we have the little sacs that can fill up with air, the alveoli. So it kind of reminds me of that. So that's how I always remember that they have these little sacs beneath their plasma membrane. Some of them are empty, but sometimes they may have cellulose in them. So they are known, the alveolates, we have a couple that we actually have three that we're going to study. We're going to study ciliates, then we're going to go to dinoflagellates, and then we're going to talk about AP complexans, and discuss them. So ciliates, the name just pretty much tells you that they have cilia. And if you remember back to biology or dual credit biology 1, we know that cilia are structures on a cell that are very short, hair-like projections that will beat in unison that allow that cell to be able to be motile. So one of the most common ciliates is the paramecium, and I've put a video in the Google Classroom. It's a very dramatic video that shows paramecium and some of the things that they do. And I like it because it allows you to see the paramecium moving, but it also is going to talk about some of their more complex structures and some of the things that they do. A few things that I want to talk about though when it comes to the paramecium is that you need to know that paramecium have two nuclei. They have one big nuclei that's called the macronucleus, and that's the one that pretty much controls its day-to-day functions. Everything that it's going to do, that macronucleus is telling it what it needs to do. And that particular nucleus will divide through mitosis, asexual reproduction. However, there is another nucleus that's found in paramecium that's smaller, so it's given the name micronucleus. And this one's pretty interesting because what happens here is this nucleus is used during sexual reproduction. So what happens is the micronucleus will form gametes, and then another paramecium they'll swap those gametes, and then that gives you a mixed genome for the macronucleus of the new paramecium that's formed. So that's fantastic because we know from genetics that you need that genetic diversity in order for it to be for it to survive in the environment. So on page 344, it goes a little more in depth into the paramecium, some things that you might want to look at. Some paramecium, graze and eat chloroplasts or algae, and then they retain the chloroplasts, and that allows them to be able to go through some autotrophic type tendencies. There's several different things about how they live, how they reproduce in that particular section, as well as in the video that you're going to watch about paramecium. This is also going to be one of the protists that's going to be researched by one of you in class for your project. So I'm not going to dive too deep into the paramecium other than to tell you these few things. I do want to tell you there is one ciliate that your book mentions that causes something called B. coli cyst, which is, I think E. coli, and it kind of does the same things. Basically, this is a ciliate that is found in some cattle. and it helps them to break down food in their gut. And what can happen is this can be transferred to humans, and usually as a dormant cyst. But of course, we know that if it gets into us, that cyst will become active. And when that happens, it can cause some gastrointestinal difficulties. So the next alveolate that we're going to look at is the dinoflagellates. If you look at that di, to me, that tells me that it's going to have two flagella. And this, because it has the two flagella, it's kind of like someone in a canoe or two people in a canoe and they're both rowing against each other. It kind of causes them to spin. So they have this kind of a rotation that we see. These are mostly photosynthetic, but something very interesting about dinoflagellates is that they are the source of bioluminescence in tropical waters. And there's a lot of speculation of what bioluminescence is for. I know I've watched a couple of TED Talks where marine biologists have discussed this. It's thought that for one, it's used to ward off predators. That could be one thing. And then there's another scientist that I saw that kind of helped to form a colony type structure. They were attracted to that light from one another. But it's neat how they form that bioluminescence. It's a redox reaction inside of them, and the byproduct is this light. And you see it a lot in tropical places. I've gotten a chance to see it once when I was traveling in Australia. I was able to see a bioluminescence group of protists, and it was pretty neat. Now, another thing that dinoflagellates are responsible for are algae blooms. One is known as red tide, and you may have heard of it before because we see it a lot of times down here in the southern United States, usually around Florida, the coast off of Florida. I've put a link to an article and a little video clip that kind of explains the red tide to you, because this is something that could affect you later on. And I just thought it was neat that you have this in your chapter. And I think it's important that you learn a little bit about it. So basically what happens is, an algal bloom is just basically when algae are going through this increase in their population, this reproduction. And so what happens is the protists, because they're putting off this, it's enriched nutrients, and it's like a runoff that you see, and it's got millions of these algae cells, these red algae cells. And so the water can get this reddish tint to it. So why is that a problem for us? Well, if you take in shellfish or other, fish that may have fed on that red algae, there is a toxin that is produced that can actually cause some issues with you. And it's a shellfish poisoning. And it's not actually the shellfish that's making you sick. It's what that shellfish had ingested. And so it's like a neurotoxin that it will develop. And some people have gotten very, very, very sick from it. And that's why sometimes people tell you to be very careful eating like raw fish or, raw oysters, because you have to be careful of those algal blooms. But we have a pretty good, group that monitors that and makes sure that people that are in the food industry know that it's happening. So you're always warned of it. So here's a picture of the paramecium. Please, please, please go and watch that video about the paramecium, because it's just phenomenal. It's so cool to watch them move around and see what's happening inside of them. Also, you can see now why it's called a ciliate. If you look around it, you see all those little cilia that are everywhere. And of course, those are what are moving to allow that paramecium to be mobile. And then here's your dinoflagellate. You'll see the two flagella, one here on the right and then one little one at the top. And that's what causes it to do that little spin. And this is one of the ones that we find with the algal blooms. So, all right. So let's go on and talk about the last the last little part of 21.5 under alveolates, and this is the apicomplexans. These are animal parasites. And they are given their name because basically it's a bunch of microtubules and secretory vessels that are found at the apex of the cell. And so that's how they were given their name. Probably the most difficult thing to understand about apicomplexans is how they go through their life cycle, because they do this switch from both asexual and sexual reproduction. And we actually talked about one of these at the very beginning of the unit when we discussed malaria. And if you'll look in your book on page 346, you'll see a breakdown of how this life cycle works for the apicomplexan plasmodium that causes malaria. So you'll see that they have it kind of written out for you there on page 346 at the top. So what happens is first, a mosquito, which we know causes malaria, bites a human. And when it bites, mosquitoes inject some of their haploid cells into the blood, and those cells are called sporozoites. And then when they're in there, they're then sent to the liver. Now, in the liver, those sporozoites, they reproduce asexually. So they go through asexual reproduction, and then they mature into another type of cell that is known as merozoites. Those leave the liver, and some of them infect red blood cells. When they get into the red blood cells, this is where they switch to sexual reproduction. And then other ones, they just become gametocytes. So when a mosquito then bites that human, another mosquito, it sucks up the blood that has that gametocyte in it, and that goes into the mosquito's gut, matures into a gamete, and forms a zygote. And then that zygote is what the sporozoite is that then gets injected into the next victim of that mosquito. So it's a really neat switch between asexual and sexual reproduction. And that page 346 has a really good picture of it. And then it has the steps of that life cycle. Another one that's really interesting is the alveolate that causes toxoplasmosis. And I did a research project on this when I was in college. Toxoplasmosis is not a very common disease. We don't see it very often because it's one that we're able to fight off pretty easily. Individuals who develop toxoplasmosis are typically someone who is immune suppressed. So it's something that we see in individuals who are suffering from HIV or AIDS, or other individuals who may have some type of immunosuppressant disease or on some kind of immunosuppressant drugs. So the big thing about toxoplasmosis that you have to be concerned about is that it's a protist that will can cause problems for women who are expecting. So women who are expecting are told by most doctors to try to limit their interaction with cats, especially cat litter boxes, because cats that maybe spend some time outdoors can pick up the cysts of this protist, and then that can be sent or can be passed through the cat into their feces. And then when you go to clean the litter box, you can inhale that cyst, and then it goes into your body, and it causes some serious problems for the fetus. Some severe neurological issues. It can destroy the brain and how the brain is functioning, and it causes a lot of problems. But that is an apicomplexan as well. And all of these are part of the A for the SAR supergroup, and that A stands for alveolates. The last thing that we're going to talk about, and here's the picture right here on the plasmodium. The last thing I want to talk about is 21.6 Rhizarians, and this of course is the R for the SAR supergroup. We're not going to spend a lot of time on this, but there's a couple things that I wanted to point out to you. So Rhizarians are single-celled. Most of them are marine. They are heterotrophs, and they have a protective shell, much like what we saw with the diatoms. In the Rhizarians, we have two groups. We have what's known as the Foraminifera, and then we have the Radiolaria. So the Foraminifera, their shell is made of calcium carbonate, and most of them live on the seafloor. And then when they die, they will be deposited on the seafloor. Radiolera does that as well, except their outer shell is a kind of a glassy silica like the diatom shell, and it has these little spikes on it. So if you look, I put pictures. This is the Foraminifera, and then this is the Radiolaria. So you can see the Foraminifera kind of has a little bit more of a rounded, whereas the Radiolaria, it's like these really needle-looking projections. So what's interesting about these is that when they die, their shells are deposited on the ocean floor. And then, over time, of course, the earth has changed. So there are some areas on the earth where we make use of these remains of the Foraminifera and Radiolaria, and those will be chalk and limestone. They have actually determined that the stone that was used for the pyramids in Egypt are largely made of shells of these Foraminifera, which probably were shed on the ocean floor, 50 million years ago. And so that's pretty interesting to me. So when you find chalk or you find limestone, it's going to be a combination of this Foraminifera and Radiolaria. And so this has been going on for 500 million plus years. So there's countless of these shells that are found on our ocean floor, and then of course in areas where there once upon a time had been ocean. So that's going to wrap it up for the SAR supergroup. We're going to finish up the last two parts of the phylogenetic tree for protist in the next lecture. So I suggest that you go through and make sure that you read these sections in the book. You can go back and re-watch the lecture. And then there are tons of resources available out there to you about some of these more specific protists that we've discussed. And I highly suggest that you go watch the video on paramecium, as well as read the article and watch the video on the red tide. And if you have any questions, please feel free to email me.